along with our moderator, Professor Barry Halliwell, to the center of the stage, where we will have a short Q&A session. For those of you who... Can I have the microphone? This one as well? Okay. Right, for those of you who do not know Professor Halliwell, he's the chairman of the Biomedical Research Council in A*STAR, and until recently, the deputy president of Research and Technology at NUS. Professor Halliwell is an internationally acclaimed biochemist who's known for his work on free radicals and antioxidants. I'll now pass the session to Professor Halliwell.
Well, we've heard two really fascinating talks about the origin of life. I personally had never thought about the genetic code or metabolism or clays in that kind of way before, so my mind has been opened. Who'd like to present the first question? Yes, please. Yeah, let's go ahead.
Hi, thank you. My name is Ajay. I'm from Yale and Hughes College. I have two questions, one for each of the speakers. So, Professor Hartman, I was wondering if you could comment a little bit more about your epiphany and tell us specifically about how the phobicity of the amino acid works out when you rearrange your columns.
How they what?
The hydrophobicity.
Oh, they were the same. I didn't change anything. I never changed the hydrophobicity, except the hydrophobicity, none of those changed at all. The columns were exactly intact, except for the change of arginine for diaper. That's all I did. I mean, this was, how should I say it, a gift from the gods. That means this is not sweating over laboratory stuff. This was, why the hell did they write the code the way they did? You know, why UCAG? I mean, I wish I would know why they did that. And then essentially just one night I had a headache and I just rearranged the code table. And the rest of my life was now, I don't know, wasted or at least amused.
Thank you very much.
You had
another question.
Yeah, quickly, please. Yeah.
Yeah. So, Professor Szostak, one of the recent discoveries has been about the bacterial fossils, which seem to go back to about 3.7 to 4.2 billion years, which just happened like a couple of weeks ago. So, my question to you is that because there's a hypothesis of exobiology where perhaps the life actually didn't have to originate on Earth, so would it matter if we know exact conditions about the primitive Earth if it were to be the case that the life actually originated outside?
Yeah, I mean, I think the idea, current thinking is in the early stages of the solar system that material was extensively transferred between Mars and Earth and possibly Venus. So within that area, I think life could have started anywhere. I would still go with Earth as the most favorable environment. I think transferring from some other star system is extremely unlikely. And, you know, no matter where life started, we have to understand how it started, right? So I don't think it really helps much to invoke just going back to starting somewhere else.
Yes, please.
I'm particularly interested by your description of the incorporation of phospholipids into the membrane. Mm-hmm. Of course, we need to have fatty acid chains of equal length. And I wonder whether you have thought about how the length of the fatty acids became to be so controlled.
I don't think we're really anywhere close to understanding questions like that because we don't have a very good understanding of how the acyl chains are generated in the first place. I mean, there are two predominant hypotheses. One is Fischer-Tropsch type chemistry in a hot, deep, high-pressure environment. The other is oxidative cross-coupling chemistry catalyzed by transition metal ions. Neither of them would give particularly homogeneous chain lengths. So yeah, so I don't think we're close to answering that.
Okay, thank you.
Next question, please. Oh, yes, David.
I mean, right at the start, and I guess it's the central question in a way, is, the chance that such an event could occur at some frequency in different planets in the cosmos. And I guess from both of your consideration, I mean, you talked initially about the potential for a choke point, some particularly difficult transition that would reduce the frequency of these events. And I wondered if you could both comment as to what in your own versions of this process you see as perhaps the critical choke point, the thing that would reduce the frequency with which these events would occur.
There was no choke point. The heart of the problem of the origin of life is really a planetary question. It wasn't something that had happened in a unique place. It happened as the planet itself evolved. Let me give you a reason why I believe that. Recently, the biggest problem in the origin of life, from my perspective, has been that 500 million years, there are no records. That means the solar system was founded at 4.5 billion. The first evidence for life that's arguable is about 3.9 billion. There's banded irons in Greenland. But the point is that 500 million years, anything could have happened. And so that's why Mars is so important. Now, why is Mars so important? Not to look for organic molecules on there, is that 500 million years, there is a record of it on Mars and not on Earth. And as I already hinted at, that era was the era of iron-rich clays, which, in my humble opinion, is life already. Those are the organisms. The question whether it will evolve into the kind of life that Szostak is thinking about, that is an interesting question. The origin of life is trivial. The evolution of life, especially in its early stages, that is not trivial. That means to look at the evolution of photosynthesis, the origin of photosynthesis, ferrous-ferric couple, CO2 fixation, that's trivial. The evolution of that simple system into this enormous system where you have Calvin cycles, NAD, ATP, all kinds of evolution of the electron donor, from ferrous to sulfide to hydrogen peroxide to water. I mean, these are the really interesting problems. And to think that the system evolved from CHO to CHOS to CHON to CHONP. Schnapps is not the right way to look at it. It started off with CHO and then moved as the various elements came into the system. So the real question is, was there a possible choke point? And the answer is almost certainly yes, because why have we lost that for 500 million years? So there is this hugely assumed that there was a big bombardment of meteorites that came at that time, which wiped out the memory here on Earth. So was that a choke point? I don't know. But it's not a choke point in the sense that a chance event occurred at the origin of life and then everything else. That's not where the choke points come. The choke points come like it does today, that you pump all this crap into the atmosphere and you got a choke point.
Okay. Well, I think it's is this obvious that we have pretty different views of how life got started on the early Earth. I used to worry that the evolution of the first RNA polymerase ribozyme was a potential choke point. I think I'm less concerned with that now, because we see a lot of small ribozymes, and you don't have to start with the replicase anyway. I think the remaining possibilities are that the geochemical situation that gave rise to all of the building blocks might have involved sort of a series of different environments forming, and maybe there's something in there that could be a rare event. You know, I have to say that as we've worked on many of these sub-problems, a lot of things I thought we had no idea of how they could ever be solved. They turn out to be trivial. So I am gradually coming around to the view that maybe all the steps actually will turn out to be easy, and there is no choke point, and life will be abundant. But you know, that's just an intuition. We'll have to see.
Any further comments?
I'd like to make a comment on that. Right now, NASA has put out a project saying what is the real problem, the origin of life? Is it the RNA world, or is it metabolism? I would like to hear from Szostak how he feels about that dichotomy.
Okay. Yeah, I think that once you have the RNA world, which certainly included prebiotically formed peptides doing lots of interesting things, I think once you've got that, the evolution of metabolism will be pretty straightforward.
Well, the interesting thing about that comment is that there is a huge school that puts metabolism first. They don't believe that it came after the RNA world. It came before the RNA world. And that's a debate. It's an interesting debate. And of course, I belong to the metabolic system coming before the RNA system. And I think as Szostak finds out all those easy steps, he's coming around close. I mean, he's beginning to believe in peptides. Now, where the hell are they coming from? And there are peptides. I think that one of the, how should I say it, arrogance of the RNA world is such that, I'll give you an example. They had this meeting. You must admit, it was an interesting meeting. Not necessarily true, but an interesting meeting about the evolution of the translational apparatus. I say, until our friend here gives us a good evolution of the translational apparatus and the genetic code from the RNA world, I will listen to them more carefully.
Any final comment before we move on to the next question?
I'll pass on.
Okay. Yes, sir.
There's a question for Professor Hartman about the thioesters here. So according to your theory, the phosphates came later. For the thioesters came first, and
yes,
then the phosphates came later. My question is, what's the selective advantage of the phosphates? We know that the phospholipids in the first talk, we can see an advantage there. How is it possible they become such an intimate part of DNA and RNA so late?
Well, first of all, I don't believe, along with my friend Dr. Szostak, that membranes are based on fatty acids. Because we have a bunch of experiments with polyglycine. And if you put a positively charged molecule on polyglycine and a protective group, 5 glycines give you beautiful bilayers. So the fact that lipids can self-assemble into bilayers polyglycine with a positive charge will do the same. Now, when you look at the evolution of membranes, membranes are made up of two things: proteins or peptides, and lipids and phospholipids. So why did it start off with peptides and that the lipids and the phospholipids came later? It's an interesting sort of new perspective, that peptides. So let me give you a little sort of feeling about, given the monomers, what is the problem that you talk about? What is the hard problem? And that's polymerization. How do you polymerize the monomers? How do you polymerize peptides, amino acids, or nucleotides, or whatever? Now, if you look at the polymerization, there's a very interesting point. If you do it with lipids, it's all sulfur chemistry. It's polymerization of acetyl-CoA. And that's how you make fatty acids. If you look at the peptides that are made in antibiotics, it's all thioesters. Now, if you look at the sugars, and the early sugars, they're all phosphodiester methods. And then you have the nucleotides is also a phosphodiester system. The interesting point is that the one place where these two polymerization systems meet is in the amino-acyl tRNA synthetases. And the question is, which way did it go? Did it start off with thioesters and move into the phosphate system? Or did it start in the phosphate system and move into the thioester system? And the answer from what we did in the phosphate-free system would say it went from the thioester into the phosphate.
I mean, with respect to phospholipid chemistry, it's quite interesting. There are two biological pathways, right? One, they both start with a lysophospholipid, and that is then joined using an activated fatty acid. And in one pathway, it's activated as a phosphocarboxy anhydride. In the other pathway, it's activated as a thioester. I mean, there's a lot of interesting ideas now about how sulfur came into prebiotic synthesis. And I, certainly think thioesters were important early on, but, there are good reasons why most, by a lot of core metabolites are phosphate esters, for example, in glycolysis, so that in an early cell with permeable membranes, if you're charged enough, you can hold on to those metabolites, right? So phosphate esters are a good way of doing that. Whereas thioesters being uncharged would let things leak out. So I think, phosphate chemistry was there from the beginning and an important part of the origin of life.
Yes, please. Yeah.
Just a question for Professor Szostak. So just going on with your RNA and lipid world and your early vesicles that would grow by osmosis, or this osmotic pressure that would cause them to swell, what's the implication of RNA polymerization on the osmotic pressure within those as a driver for the growth of these things and diffusion of RNA and molecules across this? Do you think RNA polymerization caused this, was a driver in a sense for this by polymerizing?
Yeah, actually what I was trying to say is that was this osmotic pressure-driven growth, that was our first model, but we had to reject that because we couldn't make that compatible with division. That's when we moved on to these different modes of growth that give you filamentous growth. And so that basically requires that you don't have too high of an internal osmotic pressure, right? Otherwise, you would just balloon up into a spherical shape that's hard to divide.
And there's no way you can reconcile a decrease in osmotic pressure with elongated growth in one direction, or is it just not compatible?
Well, you just need to not have too high of an internal pressure.
Question over here first. Oh. Yes, please.
To Professor Hartman. So in the Venn diagram, where you compare the ribosomal protein overlap in the three kingdoms, you hinted that the most conserved part is also like the center of the onion in terms of being early in evolution. Is that right?
Say that again. I didn't discuss the onion with respect to the proteins that universal. I didn't tell you which one. What is becoming clearer is that the peptides that are involved closer to the PTC are more primitive. And as you move out from the PTC to the periphery, there seems to be an evolution of the proteins.
Mm-hmm. So when you talk about more primitive, then you mean the building blocks that they are made of?
Well, what I'm saying is that it's more a reflection of the amino acids that are coming in. If you want to make helices, you really need hydrophobicity. Hydrophobic amino acids are very important for making alpha helices. And if you have glycine and proline and serine, these destroy helices. You cannot make helices, alpha helices. You can make with polyglycine a helix that is exactly 3 amino acids, and also with proline. These are found mainly in structural proteins like collagen or silk. So in some sense, what it seems from the GC thing and related to the growth of proteins from the PTC site is that there is a gradual evolution of purely structural materials into things that are more catalytic in terms of the hydrophilics, and then into the much more, how shall I say it, globular form.
Okay, yeah, I understand that that's a good point from the structure point of view. But if you would have an evolutionary signal of the, let's say, the building block, if you do the Venn diagram not based on full-length protein but based on the ribosomal smaller building blocks, I'm asking it for that purpose. Could have been possible that you have a tRNA not just recognizing a single amino acid but also small peptides when there is not abundance of free single amino acids, if you have enough space in the primordial ribosome?
Well, what you're asking is something...
Like, how did the small building blocks come together? Like, how do you see this evolutionary pattern of peptides coming from different organisms?
Well, there's a number of things that I didn't and cannot discuss because I don't know. What is true is that what I have shown you is the initial stages of evolution. How this system as a whole evolved. Like, for instance, in the last thing I talked about, the evolution of the tRNA. That had to be coupled with the evolution of the synthetases because the recognition of the anticodon had to come about when the tRNA had the arm and the anticodon. So the question you're asking about is, how did this system bootstrap its way? And that is not going to be an easy system to analyze because it's going to take a lot of hard work. All I've said to you is, when it's easy, like the genetic code, or when it's easy, like metabolism, then essentially it takes a couple of days and you can basically see the pattern. What you are seeing here is pieces of the pattern, but how it all integrated, that's a harder question. I can tell you it's not just me, but Lauren Williams has published a lot of papers now on how the RNAs grew and how they ligated and the mechanism of ligation of the RNA. He also has recognized, as I have recognized, the evolution of the proteins as they go towards the periphery from the center. Now, the question you're asking is, how did this whole system come together with the tRNA, the synthetases, and all the rest? That is the next time you will invite me.
Yes, please.
Okay. I'm going to ask a non-scientific question, perhaps hopefully not irrelevant. I'm particularly interested in Professor Hartman's talk about the origin coming out of clay. If you look at a lot of religions, yeah, didn't God create human beings using clay? Is that an epiphany? May I ask both professors?
I think that was his mistake, because it is human beings that will have eventually destroyed what clays made. Clay started life, evolved it, then man came on the scene, and he's about to end it. So the point is, I think in religion, the way I usually put it is, clays created man, man created God, and God help us.
Yes, please, go ahead. We have a microphone.
I understand that genetic diversity of phylogenetic or functional is a popular indicator or proxy of microbial diversity. However, I think it's unclear whether it appears as to what extent functional diversity, gene sequence diversity, and community functioning are linked. Could Professor Szostak and Hartman express an opinion on their degree of relationship and possibly their capacity to predict community functioning? Sorry.
Okay, I mean, this has nothing to do with the origin of life.
Yeah, but,
All right, so yes, I have published a number of papers on that problem. And the question is, what's driving the microbial diversity in the oceans? That's the one I'm most interested in. And the question is an ecological question. It was once proposed by a famous ecologist. Why is there so much phytoplankton in the oceans? And the answer we came up with was that when you look at the microbes in the oceans, they have 10 times more viruses that attack them. And therefore, you set up arms races between bacteria that become resistant, then the viruses then have to mutate and destroy them. And essentially, you then are going to drive these systems insane. They're going to become very, very diverse. And diversity is caused by the interactions between these organisms of predator and prey. And that's why there's so much diversity in the microbial systems in the ocean. I don't know if that answers your question.
I think that might be one of the first times I agree with Hyman.
What? Oh, I'm leaving. My purpose is now done.
Don't just leave yet, because there are a few more questions.
No, I won't.
Yes, please, go. Who's the next question?
I mean, everything was so clear.
Okay, I have a question then. Actually, I have a question which is not mine, but Sydney's. When Sydney left because he was tired, he told me, how could Hyman talk about all these things when there was no oxygen in the beginning? So can you explain it?
Well, of course, the discovery of oxygen was one of the great discoveries in photosynthesis, because until then, the redox system, the amount of free energy available for life, was relatively small. Anaerobic systems are not as have not as much free energy available to them. So again, that's something that I spent a fair amount of time with Dr. Blankenship working on the origin of oxygenic photosynthesis and how oxygen came into the atmosphere. Now, it seems that oxygen came into the atmosphere about 2.7 billion years ago. And that was probably the one great choke point, because I would say that most of the diversity at that time was destroyed by oxygen stuff. The only thing that came out of that catastrophe were bacteria, archaea, and eukaryotic cells. That is, there was a huge diversity of microbes, anaerobic microbes, and an anaerobic biosphere before oxygen came into the atmosphere. And we are looking at, when we look at archaea, bacteria, and eukaryotic cells, a very small sampling of what the biosphere looked like before oxygen came into the atmosphere.
Maybe I can just add one comment to that, which is that before there was a lot of oxygen in the atmosphere, of course there was no ozone shield. And so ultraviolet light in the wavelength range between roughly 200 to 300 nanometers could reach the surface, where it could drive photochemistry, that was an important part of the prebiotic chemistry that gave rise to the building blocks of life.
Can I have one more question? Actually, this morning I raised this issue, the CG dominance in the DNA, and we experience still until today a CG dominance. But you explained, Hyman, that from the very beginning, in your opinion, there was CG dominance, and more precisely, a solely CG code. So what is the origin of this? Because there are so many speculations. Why CG? And why AU or T came later?
Well, I mean, I think that Szostak pointed out the simplest reason. I mean, the simplest reason is you have, when the bases come together, you have 3 hydrogen bonds with CG and only 2 with AU. And that's been one of the major causes that would have preferred CG to come first, because in some sense, the amount of errors that you would make with AU is much greater. The bounding is much weaker, and therefore the error rate would go up like crazy. Anyways, that's one of the simplest ideas.
Yeah, but I don't think you can start off with all GC, right? I mean, even now, you try to replicate an all GC sequence, it's pretty much impossible. And so I think all 4 bases were there from the beginning. In fact, if you have C, you can't avoid having U, right? Deamination will give you U. So I think there's very good reasons for thinking that all 4 were there from the beginning, and the precise ratios would be tuned by environmental constraints.
So I'd like to add a comment to that. The one thing that has come out of the RNA world that I would appreciate is, of course, the stuff that von Kudrowski and Jerry Joyce have worked on in terms of ligation and amplification of RNAs. I think this business of the hunt for the RNA replicase has not been very productive. And what I would rather think, if I look at the model, I mean, I didn't have enough time to talk about the tRNA evolution a la Rodin and Ono. It does look like it was much more these tetramers that were being ligated and could form a message. They could be ligated, and then the GCs interact and you could replicate in some sense. I would rather think that when we've worked out completely the evolution of the tRNA, I think that that's when we will then come to our brothers and sisters in the RNA world and ask for help. But until then, they should realize quite easily that the RNA world was not the origin of life. And that is what has caused them infinite pain, because they have to assume a soup that nobody else does assume anymore. They have to assume a lot of things that has been, how shall I say it, the buttress of the RNA world. The RNA world did take over eventually. And it's that interesting takeover from the clays that is going to be the real question of how membranes came in, and as Szostak showed, how clays were playing a big role in the system where you can polymerize 40 nucleotides on the surface of a clay particle. That was done by Ferris. So the point is that if only he would take the clays more seriously and autotrophy more seriously, his contributions to the RNA province would be immense.
So I think I'm starting to come to the conclusion that my experiment with clays might have been one of my biggest mistakes. In fact, for polymerizing RNA, you don't need clay. You can just take your activated monomers and freeze them, and you will have long chains if you come back in a month. I think there's no experimental evidence whatsoever supporting any role for clay. And on the contrary, there's very extensive organic chemistry supporting the transition from simple building blocks, which are basically cyanide and cyanide derivatives, up to all the starting materials of biology. This is a work in progress, but it's being extensively worked on experimentally. And this is going to give us a pathway from simple building blocks up to life that is supported by experiments and not just by thinking about it.
Well, maybe you shouldn't go to him for help. But I believe you want to make a comment?
Yes. All right. I have another talk, which starts from the Big Bang to the clay. But I don't want to make it now. There was just one comment that I made in that talk. Organic chemistry is the worst system to look at for the origin of life. Why? Because three things are true. Terrestrial planets are immensely deprived in the volatiles. That's why helium is called, it was found in the sun and not on Earth. Second point, the worst liquid medium to do organic chemistry is water. You show me an organic chemist that does this stuff in water.
John Sutherland, Matt Counter, me.
And second of all,
a long list.
Organic chemistry is the chemistry of the cosmos, just by the cosmic abundance of elements. The worst place to look at the cosmic abundance of elements is on Earth. On Earth is the worst place in the whole universe to do organic chemistry. That's why one has to appeal to something much greater, which is biochemistry. Biochemistry is the chemistry of carbon, hydrogen, nitrogen, oxygen, sulfur, and phosphorus, and transition metals, and a lot of other things in water. And that is what you should be looking at. Biochemistry, organic chemistry, you leave to the organic chemists. Because of their solvents, their livers go to hell very fast.
So, we're talking about aqueous organic chemistry that gave rise to life. And before there was life, I don't think it's fair to call it biochemistry. It's just organic chemistry that happens in water. This is a hot new area of organic chemistry.
Any last question? Yes. Oh, yes, sorry, yeah.
I have you mentioned, Professor Szostak, the conditions under which these simple forms of cells could develop, which were vents, hot vents, etc. Now these vents are still there, and assuming that the vents itself,
but not the deep sea ones. These are shallow.
You talked about the ones but these vents are still there. So assuming that that they haven't really changed, is it possible to find in this environment of these vents the very early stages that you discussed about?
No, unfortunately it's not, partly for the reason that I mentioned, which is the presence of oxygen, right? So this chemistry can't go on anymore. And the other problem is that the planet is extensively contaminated with life. There's no place where you can look for primitive forms of life because, advanced life would eat it up or eat up any of the building blocks that were made. But yeah, so there's two reasons why that you can't, that's not going to work.
The reason that these vent idiocies have been propagated is that they need the enzyme which makes ATP from proton gradients. That enzyme is the most complicated enzyme that we have on this earth. And they now need that to make ATP.
So we're agreeing again. This is very dangerous.
Szostak, you've blown my mind. I need an aspirin, quick. I might come up with another GCAU code. Every time I get a headache, that's what happens.
Well, maybe you can do that over dinner. So I think this has been a wonderful exchange following a wonderful pair of lectures. So let's give our two speakers a round of applause. And now I'll hand over to Ben Seat to close.
Yep. While we grapple over the debate of clay versus RNA, biochemistry versus organic chemistry, I'd just like to highlight the next event. So 10/8, we will be talking about two things. One, about the evolution of the first vertebrates and how the first tetrapods came from the sea to the land. And secondly, how the first nerve nets formed the first primordial brains. This will be held on the 10th of April at the Biopolis. So I hope to see some of you there. Thank you.
I'd actually just, in closing, like to thank our speakers, but also A-STAR and NTU Paralemurs Institute for making this evening possible and giving us a venue. Thank you very much.